if you've ever looked at a medical diagnosis, you probably have this, I don't know, this deep-seated expectation of precision.
Oh, absolutely. Like it should be engineering.
Right. Exactly like engineering. You break your arm, the x-ray shows that jagged white line, and the doctor just points at it and says, well, there it is.
Broken or not broken, it's a clean label.
Yeah, and honestly, that is incredibly comforting. We really like our problems to be visible and neatly categorized. But then you step into the world of modern oncology and pathology, and suddenly that comforting x-ray machine feels like it is completely malfunctioning.
Yeah, the landscape gets incredibly murky very fast.
It really does. So, welcome to the Digital Pathology Podcast, Trailblazers. Today, we are throwing out the old rulebook. We're exploring how modern oncology is completely rethinking the way we look at tumors.
Moving away from those outdated, comfortable yes or no labels.
Exactly. Moving toward continuous, highly detailed spatial maps. And to guide us today, we are looking at a really brilliant new paper by Nick G. Reitsam. It was recently published in PLoS Medicine.
Yeah, it's titled Spatial Omics and AI for Clinically Actionable Cancer Biomarkers. And it's just a fantastic piece of literature because it attacks this core problem we face in the clinic every single day.
Right, so let's set the stage for you. Right now, cancer care relies heavily on this deceptively simple question. Is a tumor positive or negative for a specific biomarker?
Just a single binary label.
Yep. And that label essentially decides if you, the patient, get life-saving targeted treatments. I'm talking about therapies like antibody-drug conjugates or ADCs.
Which are fascinating. Let's actually break down what an ADC is mechanically, because it really highlights why this positive or negative label is so crucial.
Go for it.
So an ADC is essentially a microscopic smart bomb. You have an antibody, which acts like a homing pigeon, right? And it is engineered to seek out a very specific protein flag, an antigen, on the surface of a cancer cell.
Okay.
And attached to that homing pigeon via a chemical leech that we call a linker is a highly lethal chemotherapy drug, the payload.
Okay, let me make sure I'm tracking. So the homing pigeon finds the flag on the cancer cell, it attaches, the cell absorbs it, and then boom, the chemical leash breaks, detonating the chemo bomb directly inside the cancer cell.
Exactly. That is the entire promise of precision medicine. You get greater destruction exactly where the target is and significantly less collateral damage to your healthy tissue where the target isn't.
Makes total sense in theory.
In theory, yes. But here is the central issue we're unpacking today. That simple positive or negative assumption, like the idea that a tumor simply has the flag or doesn't have the flag, is fundamentally failing in clinical reality.
Because tumors aren't just uniformly painted walls, right? They don't all fly the exact same flag at the exact same height.
Not at all. They are heavily heterogeneous. So when we give a targeted therapy like an ADC, the drug's effectiveness doesn't just rely on the mere presence of a target somewhere in your body.
Right.
It relies on exactly where that target is expressed, at what specific volume, and in what surrounding microenvironment.
And traditionally, our diagnostic approach has been, well, pretty brutal, honestly. We've essentially been mashing the tissue together to find an average.
The bulk assay approach.
Yeah. Using a traditional bulk assay is like taking a blender to a complex, beautiful mosaic, pouring out the colored dust, and then trying to guess what the picture was. You know what colors are in the mix.
You know there's red and blue in there somewhere.
Right. But you have destroyed the entire structure.
Which is a massive problem. Because in cancer biology, the architecture is the vulnerability. When you use a bulk, average-based test, you completely gloss over the underrepresented regions of the tumor.
Hiding spots.
Exactly.
Yeah.
And those tiny specific niches, the little clusters of cells that maybe didn't get ground up perfectly in that metaphorical blender, those are often the exact regions responsible for tissue invasion or metastatic seeding or a relapse years down the line.
Oh, wow. So the blender approach actively hides the most dangerous parts of the cancer.
It really does.
But, I mean, if we don't average it out, if we don't just blend it up to get a baseline reading, how do we actually map something that complex in a clinical setting? I imagine pathologists don't have all day to stare at one slide and count every single cell.
No, they definitely don't. And this is where Spatial Omics comes to the rescue. The paper dives into tools like spatial transcriptomics and multiplex spatial proteomics.
Okay, spatial transcriptomics, what's the mechanism there?
So it basically lays a microscopic grid over an intact slice of tissue. Instead of grinding the tissue up, the machine reads the RNA, which are the genetic instructions the cell is actively using, right where the cell sits on the slide.
Wow.
Yeah, it works at a single cell resolution.
So it's geography. We're mapping the terrain instead of just taking a census.
Exactly. We can interrogate exactly where target-expressing cells live. We can see how they interact with their immediate cellular neighborhood. And crucially, we can see if there are reservoirs of target-negative cells hiding out in the tissue.
Just waiting to seed a recurrence after the therapy successfully kills off all the positive cells?
You got it. Reitsam's paper actually highlights some fascinating specifics here regarding recent spatial atlases of brain tumors, both in adults and children.
Right. They looked at antigens that we actually try to target with drugs, right?
Yes. You'll hear the alphabet soup in the literature, things like B7H3, EGFR, or IL-13RA2.
But for our trailblazers listening, just think of these as different types of molecular antennas sitting on the outside of the cancer cells.
A perfect way to visualize it. And the spatial maps reveal that these antennas might only exist in a tiny, isolated subpopulation of the tumor cells.
Which you totally miss in a blender.
Completely. And if we connect this to the bigger picture, it's not just about finding those antennas on the cancer cells themselves. It's about the physical interactions between the cancer cells, the immune cells trying to fight them, and the stromal cells.
Which are basically the structural support cells of the tissue, right?
Yes. And depending on how those cells are physically arranged in space, those interactions can either help your immune system attack the tumor or
they can build a literal fortress.
right, a physical and chemical fortress that helps the tumor evade the attack entirely.
Okay, so we now have the biological technology to create these incredibly complex, beautiful spatial maps. We can literally see the fortress, but reading the paper, that seems to lead us straight into a massive logistical wall.
The data bottleneck.
Yeah, generating one beautiful map in a heavily funded research lab is one thing, but how on earth do we make this data reproducible and scalable across different community clinics, different hospitals, and over, different time frames?
That is what we call the pre-analytical problem. And it is a logistical beast. Before a slide even gets placed under a microscope, it has to be physically prepared. The tissue has to be fixed in chemicals to stop it from degrading. It has to be stained with specific dyes so we can see the structures, and it has to be scanned into a digital format.
And every single one of those manual steps introduces variation.
Every single one.
Right, because a lab in New York might leave the tissue in the fixing chemical
For an hour longer than a lab in London.
Exactly.
Or different digital scanners might interpret the color purple slightly differently.
Like how two different TV screens look different in a living room.
Perfect analogy.
Yeah.
Add to that the human element. The inter-observer variability is huge.
You mean two doctors disagreeing?
Yeah, two brilliant, highly trained pathologists might look at the exact same stained slide and give two slightly different categorical scores. It's not a lack of skill, it's just the fundamental limit of human subjectivity when you're trying to quantify thousands of microscopic data points by eye.
And this brings us to artificial intelligence and computational pathology.
Yes, the computational cure.
AI isn't just coming in to do the same job a human does, just faster, right?
Yeah.
It steps in to estimate continuous measures. Instead of a human squinting and saying, this looks like a category 2 positive.
The AI says exactly 43.2% of these cells are positive, and here is the exact gradient of intensity across the entire tissue section.
It replaces subjective, chunky categories with scalable, quantitative data. And we're already seeing this roll out in the clinic.
We are. The paper notes how quantitative computational scoring is currently being used for the PD-L1 biomarker in non-small cell lung cancer.
And PD-L1 is that protein that basically acts as a stop sign for the immune system, telling it not to attack.
Exactly. So measuring exactly how many stop signs are present is crucial for deciding if immunotherapy will actually work.
Another great example from the sources is TROP2, which is another target for those ADC smart bombs we talked about earlier.
TROP2 is notoriously tricky.
Yeah, the physical staining for it is heavily heterogeneous. It's incredibly patchy. And furthermore, the stain mostly localizes right on the cell membrane, the ultra-thin outer skin of the cell.
Right, rather than lighting up the whole center of the cell.
Exactly. So using traditional human-estimated cutoffs to judge a faint ring of color around a patchy cluster of cells, that's an incredibly fragile diagnostic method.
It's genuinely unfair to ask a human visual system to accurately and consistently quantify thousands of faint membrane stains across a massive tissue slide.
Day in and day out without fatigue.
Right. AI algorithms simply don't get eye strain. They look at pixel intensity mathematically.
But I want to push back or at least clarify something here, because this is where a lot of professionals get nervous. Are we just using AI as a hyper-fast calculator to count cells, or is the AI actually extracting biological meaning that a human pathologist simply cannot see, no matter how much time they have?
What's fascinating here is that it's absolutely the latter. AI is seeing patterns humans fundamentally cannot process. And that brings us to the bleeding edge of this entire field.
Virtual omics.
Virtual omics.
Okay, break the mechanics of this down for me. What is it?
AI models are now learning to infer high-dimensional hidden molecular information directly from routine, everyday H&E stained slides.
H&E. That stands for hematoxylin and eosin. For any trailblazers who haven't looked through a microscope recently, these are the standard pink and purple slides that have been the absolute bedrock of pathology for over 100 years.
They really have.
The hematoxylin dye binds to DNA and turns the cell nucleus purple, and the eosin dye binds to proteins and turns the rest of the cell body pink.
That's the basic chemistry, yes. But to a human, it's just pink and purple shapes. But these AI models are being trained on paired data sets.
Okay, meaning what?
Meaning the computer looks at the standard pink and purple H&E slide, and then it looks at the highly complex, massively expensive molecular data for that exact same piece of tissue. And over millions of iterations, the AI learns the hidden mathematical associations.
Wait, so the AI realizes that a specific microscopic change in the texture of the purple nucleus, combined with the slight change in the geometry of the pink space between the cells things a human eye just perceives as a blur, actually mathematically correlates with a specific genetic mutation.
Precisely. It predicts the underlying transcriptomic or proteomic reality just from the physical shapes and textures on the slide. It predicts the unseen.
That's wild.
It is. The paper actually highlights an incredible recent study in lung cancer where this was put to the test. The AI combined standard H&E morphology with virtual spatial proteomics to predict whether a patient would respond to immunotherapy.
And the results were staggering. It outperformed the clinically established biomarkers.
It did.
The AI's prediction off a cheap pink and purple slide was actually better at predicting patient survival than doing a costly physical test for PD-L1 expression or measuring the tumor mutational burden.
Which involves physically counting how many genetic mutations the tumor has. It is a total paradigm shift. We're extracting $100,000 worth of molecular insight from a $3 piece of glass.
But this raises a massive interpretive dilemma, and I really want to zero in on this because it's a fascinating clinical gray area mentioned in Reitsam's paper. The sources note that the actual statistical correlation between these virtual AI predictions and the true physical protein expression in the tissue is often only moderate or even weak.
Yeah, that's the catch. The morphology-based approaches have ceilings. If a protein marker doesn't physically alter the shape or texture of the cell in a way the AI can mathematically detect, the prediction of the specific protein level falls apart.
So here is the clinical dilemma for you, trailblazers. Let's say an AI looks at a standard H&E slide and accurately predicts that you will survive, or that you will respond beautifully to a specific drug.
The math says it works.
Right, but when we actually grind up the tissue and physically test it, the biological mechanism, the protein the AI said was driving this, isn't actually there at the levels the AI predicted.
The prediction is right, but the biological math is wrong.
Exactly. Should we trust it? Do we make life-altering treatment decisions based on a black box that gets the clinical outcome right, but the biological mechanism wrong?
It forces the clinical world to ask what we value more, mechanistic truth or predictive power. If the virtual assay accurately predicts clinical benefit, which is what you and your doctor actually care about, should it be integrated into the workflow, even if we don't fully understand how it arrived at the answer?
It's a tough call.
It is. The paper heavily suggests that while this predictive power is incredibly promising, it requires deep critical validation. We cannot blindly abandon biological reality just because an algorithm boasts a high survival prediction rate.
We ultimately need to know why a treatment works. Otherwise, we're flying blind when the AI eventually makes a mistake.
Absolutely.
But, to ground this whole discussion in immediate clinical reality, we don't even have to look far into the future. We can look at breast cancer right now. Therapeutic advances are actively breaking our existing human-driven biomarker frameworks today.
Oh, this is playing out right now with the HO2 reclassification crisis.
Let's look at the DESTINY-Breast06 trial. This is a perfect example of pharmaceutical technology outpacing human diagnostic capability.
It was a massive phase 3 trial involving that ADC smart bomb we discussed earlier, specifically one called trastuzumab deruxtecan, or TDXD.
Okay.
Historically, the HR2 protein was treated as a strict binary. Your tumor either had a massive amount of HR2 antennas, meaning you were HR2 positive and got the drug, or you didn't, meaning you were HR2 negative and got nothing.
But TDX is such an effective smart bomb, and its chemical leash is designed so perfectly that it has something called a bystander effect.
Yes.
When it detonates inside a cell with the HR2 flag, the chemotherapy leaks out and kills the neighboring cancer cells too, even if they don't have the flag. And because of this, the drug worked for patients classified as HR2 low.
And the data went even further. The trial showed exploratory but directionally consistent benefits for patients with HR2 ultra-low disease.
Meaning they barely had any flags at all, but the drug still found a way to work.
Exactly. Suddenly the binary is completely destroyed. Pathologists are no longer just looking for a clear positive or negative. They are forced to distinguish between a cell being HER2 zero, meaning absolutely no flags, and HER2 ultra-low, meaning maybe a tiny, faint dusting of flags.
Which is asking a human eye to do the biologically impossible.
It really is.
Think of it like this. It's like asking you to distinguish between 50 different shades of gray with your naked eye under varying lighting conditions in different rooms versus just using a calibrated digital light sensor.
And the clinical data backs up that analogy perfectly. At these critical, low-expression thresholds, the boundary between zero and ultra-low human agreement drops to 70% or lower.
Wow.
Yeah, nearly a third of the time, leading experts disagree on a classification. And that single human disagreement determines whether or not a patient receives a highly effective, life-prolonging drug.
That is a diagnostic coin flip you do not want to be on the wrong side of.
Definitely not.
But what happens when you point AI at this specific physical problem?
When we use AI-based immunohistochemistry analysis, it has demonstrated a pooled sensitivity of about 97% for identifying patients eligible for TDXD.
It only misses about 3% of eligible cases.
Exactly. The human visual limit is clear, and AI precision easily surpasses it at these borderline decision boundaries because it is counting pixels, not making visual estimates.
So what does this actually mean for the clinicians sitting at the desk trying to treat you? Reitsam paints a picture of the future pathology report, and it is fundamentally different. Instead of a piece of paper that just says HER2 low in bold font, the clinician receives a comprehensive spatial phenotype.
The report of the future reads like a meteorological weather map. Instead of a single word, it says 15% of tumor cells show weak but mathematically detectable expression. Furthermore, these specific cells are clustered in a stromal-rich, immune-excluded region of the tumor.
That is continuous data. It doesn't just tell the doctor the target is physically there. It tells them if the physical drug can actually reach the tumor.
Or if it's going to be blocked by that physical stromal fortress.
Right. It tells them if the immune cells have access. It tells a complete dynamic biological story.
Okay, but wait. If continuous spatial AI tracking is the undisputed future of oncology, we have to talk about the massive elephant in the room.
Cost and equity.
Cost and equity. Because right now, you're describing a future where we need gigabytes of data and supercomputers just to read a single tissue slide.
Yeah.
How on earth does a rural community hospital afford that?
The equity challenge is brutal. High-cost spatial platforms often exceed $250,000, $250,000 per single instrument. And that makes sense when you look at the mechanics.
Yeah, these machines use complex cyclic immunofluorescence or bar-coded molecular probes. It can take days just to run one slide through the machine.
And beyond the hardware, you need massive server infrastructure just to hold the sheer volume of data generated. You need dedicated bioinformaticians on staff to translate the raw data into something a doctor can actually read.
For low- and middle-income countries, or even just regional community hospitals, this is entirely inaccessible. It threatens to widen health disparities on a global scale if we aren't careful.
We can't just invent life-saving diagnostic technology and then shrug and say, well, good luck affording it. So what is the practical solution here? Reitsam actually advocates for a hybrid workflow.
He outlines a very pragmatic two-pronged approach. First, we must aggressively deploy robust, low-cost AI assays built on routine H&E pathology for the masses.
Right.
If an AI can look at a cheap $3 widely available slide and give a highly accurate quantitative readout of the tumor microenvironment, that democratizes precision medicine immediately.
We use the AI to pull maximum diagnostic value out of the cheapest, most standard test available.
Exactly. Then, on the other hand, we reserve the incredibly expensive high-dimensional spatial single-cell omics for specialized academic centers and massive clinical trials.
We use the big $250,000 machines to calibrate the AI, to track tumor evolution deeply over time, and to discover the next generation of drug targets.
It's the only way it scales.
Technology and workflows are one thing, but what has to actually change on an administrative level to make this a reality? Because science moves at the speed of light, but healthcare policy famously moves at the speed of a glacier.
The policy updates needed are massive. First, we need established reimbursement pathways for these AI assays. Right now, billing codes are built for human labor.
Interesting.
Yeah, if an algorithm provides clinical benefit and saves a patient from receiving the wrong toxic drug, the healthcare system needs a mechanism to actually pay for that algorithmic analysis.
That makes a lot of sense. What else?
We need regulatory frameworks for repeat testing, because tumors evolve. A molecular test done at the day of diagnosis shouldn't be the final word 3 years later when the cancer relapses.
Totally.
We also need multi-center ring trials, which are essentially massive proficiency tests across different global labs to ensure that an AI reads a slide the exact same way in Berlin as it does in Boston or Mumbai.
And most importantly, equity guardrails.
Yeah.
We need access benchmarks actively built into the rollout of these technologies to prevent the widening of health disparities.
Ultimately, the goal isn't just to use AI because it's novel and exciting. The goal is to build a diagnostic ecosystem where highly resolved spatial biology informs what we need to measure in the tumor, and AI determines how to measure it reliably and continuously.
And that perfectly brings us to our summary of this incredible shift in modern oncology. We are moving away from static, binary labels, the simple yes or no, the broken or not broken X-ray mentality, and moving toward continuous living maps of cancer ecosystems.
It's a fundamental change in how we perceive the disease.
It changes the entire paradigm. And it leaves us with a final provocative thought that builds on everything Reitsam calls for regarding continuous and adaptive trial platforms.
Yeah, let's hear it.
If we accept that a tumor is not a static object, but a constantly shifting heterogeneous ecosystem, and if we now have the AI tools to track it continuously and non-invasively by analyzing routine slides and virtual omics over time,
Right.
when does the entire concept of the baseline snapshot biopsy become completely obsolete in modern medicine?
Wow. If the disease is a feature-length movie, why are we basing our entire life-altering treatment plan on a single Polaroid taken on day one?
That is the ultimate question we have to answer next.
It really is.
And that brings us right back to where we started. The jagged white line on the X-ray is comforting, but it's just a snapshot in time. Biology doesn't stand still, and thankfully, neither does our technology. Thank you for joining this discussion on the Digital Pathology Podcast, Trailblazers. Keep pushing boundaries, keep questioning the standard protocols, and we'll catch you on the next one.